All right, so hello everybody. Once again, I am Miguel Lujan, a third-year biochemistry major and a member of the mock entomology lab, and this is the effect of plant immunity elicitators on lettuce metabolite composition and resistance to in-patients necrotic spot virus. So moving on to the first slide, we're going to talk a bit of background on my presentation. So the Salinas Valley in California is currently undergoing an epidemic affecting its lettuce industry. The growers are suffering major crop losses due to a plant virus known as in-patients necrotic spot virus, or INSV, which is causing the lettuce in the fields to become damaged and wilted. So as you can see on the figures on the right, the first shows a picture of a plant in early stages of infection, while the second shows slightly more progressed signs of infection, which include browning and wilting. The main vectors of in-patient necrotic spot virus are thrips, which are tiny insects that feed on plants. To resolve this issue, farmers have tried targeting the vectors with pesticides. However, this is not very effective as thrips are capable of transmitting the virus to the lettuce crops before they are affected by the pesticides themselves and ultimately die. Thrips only need to feed once on a lettuce plant in order for it to potentially transmit its infection onto the lettuce, even if it later succumbs to the pesticides. There's also the consideration that pesticides are also having negative effects on the environment, which means there needs to be even less used. Overall, this is an ineffective and non-viable method for combating this issue. Moving on to the next slide, we will briefly be talking about the method of transmission for the virus in-patients necrotic spot virus. So to begin with, thrips are tiny insects which land on lettuce and begin feeding upon it. When the thrips feed on lettuce plants, they will introduce the infection inside the plants, which will begin to replicate. Then to acquire the thrips in the virus in thrips, only thrips in the larval stage of development are able to become a vector of the virus. Adults cannot become the vector even if they feed on the infected lettuce plants. However, only thrips who acquired the virus in its larval stage and have matured can transmit the disease, which in turn infect lettuce and continues this cycle. So a potential solution we can combat the spread of this virus could be accomplished through the use of plant elicitators. So to better explain, plant elicitators are chemicals that mimic signaling molecules produced by the plant during defense responses. They have been around in agriculture and commercially available and are used to activate the plant's immune system, or better known as priming the plant in order to protect the crops from disease. Prime plants are better prepared to resist infection by eliciting an early signal towards the plant's immune system before potential infection. Essentially, plant elicitators can be used to trick a plant's immune system into believing it is under attack from a virus, in which case it will elicit a defense response to keep it in a primed state. When the plant is in its prime state, it will for a short time keep its defenses up continuously to fight off infection, which is why we believe the elicitators might be a potential solution to the problem we are facing. Plant elicitators could decrease the chance of infection by strengthening the plant's response when a vector introduces the virus and decrease the damage the virus does by attenuating the infection symptoms. But making the plant defend itself takes resources, so it is important to make sure any elicitators don't have negative effects on plant growth. So moving on to the research questions we wanted to address in this project. So this study was designed to examine two things: whether elicitators might have negative effects on lettuce growth and leaf quality, and two, whether elicitators induce resistance against INSV. To answer these questions, we conducted a first experiment, which objective was to evaluate the effects of elicitators on lettuce traits important for consumers. This included effects on plant growth and metabolite composition. As for our second experiment, we wanted to evaluate the elicitators from the first experiment for effects on resistance to INSV. This included virus infection percentage of the different treatments and a virus titer levels, which was how much virus was present. So an overview of my first experiment consisted of was that lettuce was planted and grown initially in an incubation chamber with elicitators being applied 2 weeks after planting at full dose concentrations. The specific elicitators applied were Actigard, Cabral EG, DSEC, Regalia, and water for control. After elicitators were applied, the lettuce was moved into a greenhouse with a 16-hour light, 8-hour dark photo period. So to the right is some of my lettuce germinating in the growth chamber before any of the listers were actually applied. So to measure plant growth in the lettuce was harvested 3 weeks, 2 days post-application of the listers through measuring weight. We measured weights immediately to get the wet weight of the lettuce with the water and still inside the lettuce plants. Then the lettuce samples were placed in a hot box for drying for a couple of days. After drying out the water from the lettuce, the dry weight was measured. This was done in an effort to get the plant growth measured by weight in its natural state with water, and then to take the water out for its overall unaffected biomass. So moving on to the GCM analysis, we took 2 sample time points for the analysis. So the first time point was taken 1 week post-elicitor application, while the second time point was around 3 weeks post-elicitor application. Samples were collected using a hole puncher and placed in liquid nitrogen to preserve sample integrity. Samples were then homogenized, extracted, and derivatized for later use in GCMS. For this, we compared metabolite compositions of samples with glucose, fructose, and sucrose measurements. The processing and analysis of samples is still ongoing and is in work. So to the figure on the right is a picture of some of the samples in the process of extraction with the GCMS. So moving on to the methods for experiment 2 overview. Lettuce was also grown in a similar manner as before with experiment 1, with just slightly different time points. So elicitors were applied 13 days after planting at full dose concentrations. And once again, these were the elicitors known as Actigard, Caprol-EG, DSEC, Regalia, and water as a control treatment. So for this experiment, each treatment was inoculated with INSV 7 days post-elicitor application. So instead of using thrips to inoculate the plant, we had to do mechanical inoculation, in which we got previously known infected tissue and homogenized it with buffer and applied it to lettuce leaves with sea light to mimic tiny bites, which would have gotten the virus inside the plant. Samples were then harvested 19 days post-inoculation using the same method of taking leaf punches and immediately placing in liquid nitrogen to preserve the sample's integrity. Now moving on to the methods of experiment 2, lysin analysis. So elicitor-induced INSV resistance was measured through ELISA. So to briefly summarize, ELISA uses antibody interactions to measure. And in this particular process, the wells of a microplate will turn yellow if the wells in the samples have INSV inside of them. And at the end of the process, optical density of each well was measured using a spectrophotometer to see how our OD of our samples compared to our positive and negative controls. And to the figure on the right is a picture of such a microplate that some samples showing signs of INSV, hence the present yellow, and some samples that appear to not have any INSV. So moving on to the results of experiment 1 with plant growth, we analyzed the plant weight of each treatment of lettuce immediately after harvesting and found some results. Below are 2 graphs looking at how different treatments affected both wet weights and dry weights of plants. Both had treatments on x-axis and average weights in grams on y-axis. Looking at the first figure on the left, we can see the graph showing data for elicitor effects on wet weight. Here we can see that there was no significant differences between the treatments and control. We can only see that Actigard has some slightly negative plant growth, while Regalia has some slightly positive effects on plant growth. However, they are not significantly different, meaning that the p-value for the treatments did not go lower than 0.05 relative to the control. And moving on to the graph on the right, we can see the results for dry weight. And this is not much different from wet weights, as they are very similar in terms of growth. And there is also no significant differences between the treatments and the control. And the analysis of the dry weights also did not have any treatment p-value lower than 0.05 relative to the control treatment. So moving on to the results from experiment 1 regarding our plant metabolites. Below we have 3 different graphs showing averages of fructose, glucose, and sucrose from left to right respectively. These are average amounts in micrograms calculated through areas on GCMS curves. For this part of the experiment, it is still in progress. However, with the data we currently have, we can see a trend in that Actigard and DSEC appear to have the lowest amounts of metabolites, at least for fructose and glucose, while regalia has slight increases in metabolite levels in all three. So with this current trend, regalia appears to be the best in producing favorable metabolites. We can say this is the current trend. However, we still need more analysis to say that this is what we should expect for how elixers affect metabolite composition. Now moving on to the results of experiment 2's Elisa analysis. Looking at the infection rates and severity, we can see a graph depicting the infection rates on the left with treatments on the x-axis and percent infection on the y-axis. So looking at this graph, we can straight off the bat notice something immediately off. If we look at Actigard, we can see that Actigard elicitor has reduced infection percentage by nearly half of that of control, with slight decreases with other elicitors. Though DSEC also has some decrease going on, we can see that Actigard is clearly the best elicitor in reducing infection percentage, which is also what we saw with Actigard working to decrease percentage of infection with preliminary data. Then looking to the graph on the right, we can see that Actigard is also significantly reducing infection severity measured through titer. We could also see that regalia is also significantly reducing titer levels as well, even if the infection percentage from the left graph did not decrease. So from preliminary data, we can also know that Actigard did slightly decrease the infection severity before as well. So this result is semi-consistent in that regard. All right, so moving on to a general summary of my experiment. So to begin with, the elixers had little to no effect on plant growth, with only slight effects that were not significant enough. Regalia appears to have the best increase in favorable metabolite composition overall, and there was noticeable changes in plant-induced resistance to INSV. More in particular, Actigard seems to be the best elicitor for lowering infection rates of INSV with romaine lettuce, and for infection severity, Actigard seems to be the best at lowering titer levels as well, with regalia just slightly behind. So moving on to the next slide. This slide will conclude our findings. And based on our experiments and data, we found that the most promising elicitor to see stopping infection and reducing titer levels appeared to be Actigard and regalia, with regalia only significantly reducing titer levels. Regalia is also showing a slight trend in increasing favorable metabolite composition compared to other elicitors. However, further analysis needs to be done to verify this trend. So our future goals will be moving forward to test elicitors in a real lettuce field to verify our findings and continue our analysis of our elicitors. Commencing in early summer 2022, there will be field trials going on with romaine and iceberg lettuce using our findings, which will be funded by the Department of Pesticide and Regulation. We will also be testing elicitor combinations and the possibility of multiple doses and their effects on lettuce in the near future. Lastly, I would like to thank everyone in the mock entomology lab for supporting me in my experiment over the school year. I would like to personally thank my faculty member, Dr. Carey Mock, and Crystal Watrous for helping me mentor me through this process. The mock lab has been indispensable in guiding me along my project, and I could not have done it without them. I would also like to thank the California Leafy Greens Research Program for partial funding in this project as well. And here are some references I used to progress my project. And so thank you all for your time, and I would like to open this presentation up to questions now.